Hi, I'm recording this short video to give you some background on our latest production, the Cryo-EM Interview Series with Richard Henderson, and also a bit of information about the work that we do. So cryo-EM stands for electron cryo-microscopy as Richard defines it. It's a technique that uses transmission electron microscopy in combination with the samples in an environment of vitreous water at liquid nitrogen temperature, as well as sophisticated data analysis methods. So resolving the structure of biomolecules is a fundamental step for understanding how life works, and for the development of new drugs, treatments, vaccines, and so on. One recent example is the coronavirus, the COVID-19. For example, in a highly cited paper titled Structure, Function and Antigenicity of SARS-CoV-2 spike glycoprotein, published in the journal Cell in April, the authors mentioned that they determined the cryo-EM structures of a SARS-CoV-2 S component, which provides a blueprint for the design of new vaccines for the virus. Of course, there are plenty of other examples, especially after 2012, when cryo-EM became very popular. A few decades ago, resolving the structure of biomolecules at atomic level would have taken years, if not decades. It would have included complex crystallization methods and measurements using synchrotron X-ray diffraction, which is a topic we cover in our interview with Venki Ramakrishnan. So if you're interested, you can check the link below. Now, thanks to the relatively new technique of cryo-EM invented by Richard Henderson and colleagues, resolving the structure of certain biomolecules could just take a couple of days. This is why Richard Henderson was awarded the 2017 Nobel Prize for Chemistry with Jacques Debouchet and Joachim Frank for developing cryo-electron microscopy for the high-resolution structure determination of biomolecules in solution. So Richard Henderson got a PhD from the University of Cambridge in 1969. After a postdoc in the US, he returned to the MRC lab of molecular biology in 1973, where he still works today. So in our interview series, Richard is interviewed by Majd Al-Sari from the Cavendish lab in Cambridge, and we decided to split the 2-hour interview into several episodes, which will be published probably every week or so. You will find the new episodes in our YouTube channel, in particular in this playlist. So in this interview, Richard talks about the history of electron cryo-microscopy, including the discoveries made by his colleagues, Jacques Debouchet and Joachim Frank, and of course the work he did on the purple membrane. He also talks about synchrotron radiation facilities and how researchers can now access cryo-EM microscopes. He discusses the improvements that can be done to further push the resolution of cryo-EM, and explains the grand scheme towards a complete physical theory of cryo-EM. One of the issues of cryo-EM is that these microscopes currently cost £5 million or so Richard has been working towards the development of a more affordable cryo-EM. And finally, he mentions his business activities, his startups, and so on. So as I said, these videos will be published in our YouTube channel, so if you want to get notified, please subscribe to our channel. The transcripts will be also published in our Scientific Video Protocols platform, and SciViPro is the first full open access peer-reviewed video journal which publishes in 4K cinematic quality. SciViPro is published by my company, Bulaki Limited, and I decided to work on SciViPro about 2 years ago, when I realized that scientific work could have been better communicated to peer scientists with video articles, rather than simply with standard articles. Think about that. We scientists do research and write papers. Then we send these papers to journals for free, they get reviewed by reviewers that work for free, and then journals typeset these manuscripts and generate HTML pages with the PDF. And that's basically your work published. And basically we pay journals for coordinating the review, typesetting, maintaining the journal infrastructures, and maybe for their reputation. So with SciViPro we don't charge for this stuff. We just charge for the video production. You will still get your HTML page, your PDF, and your manuscript gets a DOI and then is indexed by Google Scholar, of course. So there are no subscription fees. All articles are licensed with the CC BY 4.0, which means that they can be reused elsewhere. All videos are uploaded in our YouTube channel, which is accessible to everyone. In principle, we only publish works in natural science, such as physics, chemistry, engineering, biology, and so on. We don't do anything that involves medicine, surgeries, experiments on animals, and of course we work both with academia and companies, and essentially we offer a sort of bridge between standard peer-reviewed publications and science communication. So we prefer to work on quality rather than quantity. If you watched Breaking Bad, you might remember that when Jesse Pinkman mentions that they don't need a criminal lawyer, they need a criminal lawyer. So a lawyer who is a criminal. And in our case, you don't need a scientific cinematographer, you need a scientist cinematographer. A cinematographer who is a scientist. See, the thing is that you don't want to talk to cinematographers that have no idea of how science works. You want to talk to a scientist that is a cinematographer that understands the science that you are doing and that knows how to translate these ideas into an appropriate video article. So if you're looking for quality, proper communication, reputation for your article, please get in touch. You can visit www.scivipro.com or send us an email at orders@bulaki.com. Thanks for watching.